because of my own jet lag. Today what I'll do is to tell you about some of our recent work on depression, and I'll begin by putting the work briefly in some clinical context. It's impossible to overstate the impact that depression has on humanity. It's been estimated by the World Health Organization that depression is the number 4 cause of disease burden worldwide. Number 1 in women, expected to be number 1 in men as well within the next few decades. About 5% of people in the population get depressed at some point in their life, severely depressed, and the illness is much more common in milder forms. Despite the impact that depression has, we know still very little about it. We know that a little less than 50% of the risk is genetic, but no causative genes have yet been definitively found in large GWAS studies. Role for chronic stress has been implicated in some people, but in others there's really no apparent role for stress. What we do figure is that depression is not a single disease, it's syndromic, it's a collection of many illnesses that give rise to behavioral disorders with some common symptoms. Now, despite the fact that we don't know much about what causes depression, we have some pretty good treatments. There's a range of for depression. However, all of these were developed by serendipity based on discoveries about 6 decades ago. These newer antidepressants have the same basic as those original medications. And recent clinical trials have indicated that more than half of all people with severe depression are fully treated with available treatment. So we still need to do much better. Depression, like syndromes, are diagnosed solely on the basis of behavioral abnormalities. We still have no the big field, no blood syndrome. Depression is diagnosed, like all psychiatric syndromes, from the Diagnostic and Statistical Manual of Mental Disorders, and the behavioral abnormalities are listed here. I just want to highlight a couple of things. First, that depression involves abnormalities in several domains of behavior and mood, cognition, and more physiological functions, which are called neurovegetative symptoms. But note that when you could make the same official diagnosis of major depression, whether a person eats too little or too much, sleeps, loses weight or gains weight, sleeps too little or too much, has low energy or too much energy, you know you have a problem with diagnosis, right? So that's, again, emphasizing the point that there is no single disease entity of depression. It's a mixture of many disease states. Recent work in brain imaging has identified circuits in the brain that seem to be important in controlling mood under normal conditions and may function abnormally in depression, largely validating what had been known for decades based on lesion and related studies, identifying so-called limbic brain structures important for emotional behavior. And this is a sideways or sagittal section of a human brain defining what these major brain regions are. Broadly speaking, there are large regions of prefrontal cortex, in the temporal lobe, the amygdala and hippocampus, in the basal forebrain, a region that I'll be focusing on quite a lot today, the nucleus accumbens, a major reward region in brain, and hypothalamus. And importantly, each of these 4 brain regions are innervated by the brain's 3 major monoamine pathways. Dopamine arising from neurons in the ventral tegmental area of the midbrain, serotonin from the dorsal raphe, and norepinephrine from the locus coeruleus. Now, presumably, it's the innervation of these regions by the raphe, serotonin, and the locus coeruleus, norepinephrine, which give today's antidepressants their mechanism of action, since all of today's medications act directly on the norepinephrine and serotonin systems, at least initially. In the last decade or so, there have been what I think are some of the most exciting demonstrations in psychiatry function of neural circuitry, and that has to do with deep brain stimulation. So this is actually taking patients who are so severely depressed, neurosurgical approach is justifiable, and lowering metal electrodes into certain regions of brain and stimulating those brain areas. And among many brain areas tested, 2 in particular seem to be effective in alleviating the symptoms of severe depression. And in fact, some of the sites even elevate mood in normal individuals. And those 2 regions are a region of prefrontal cortex, referred to as area 25, or the subgenual region of anterior cingulate cortex, and the nucleus accumbens. And in particular, the nucleus accumbens is the site where direct electrical stimulation elevates mood, even in individuals who are not depressed. Now, without being too nihilistic about it, I think this is really all we know definitively. There have been a lot of theories, a lot of molecules identified, but really, when it comes to very little information about the clinical symptoms. So that's where we come in. We study animal models, like in medicine, trying to advance our understanding of disease, laboratory animals. Only the main problem in psychiatry is the inherent limitation in the ability to use animal models to study the basis of mental disorders. Some of the core symptoms of all mental illness, depression included, are inaccessible in animals. And that is no doubt one of the major reasons why advances in psychiatry have lagged behind those of most other medical specialties. What I hope to convince you of today is that despite this dilemma, it is possible to make major advances by applying the tools of molecular and cell biology. So what are animal models of depression? The most widely used are referred to in the field as despair-based tests. They're listed here. They're used, I think, mainly because they're rapid and easy to perform, but they don't really make very much sense. They're not highly validated. I'll just describe one of these tests. The forced swim test is where you take a normal rodent, rat or mouse, place it in a beaker of water. The animal will struggle for a time. After a few minutes, it will stop struggling and float. Rodents are buoyant. They don't sink. They float. Investigators in the field have decided that when a rodent floats, they're despairing. I mean, that makes no sense to me. What we know is that when a rodent is given a single dose of an antidepressant, they struggle for a longer period of time. The problem is that antidepressants don't elevate mood in depressed humans except after chronic, many weeks of administration. And yet in these assays, antidepressants act acutely. Now, it is true that that all of today's available antidepressants work in the forced swim test, but it's a bit of a catch-22 because drug companies would only pursue medications that work in the forced swim test, and they wouldn't pursue others that don't. And I think I don't have to spend more time making the point that these tests in general lack etiological and face validity. So what I'll do today is tell you about a model that we've been using that we think has greater validity. It's not perfect, but it has greater validity, and then illustrate how we've used that model to derive something about the molecular mechanisms of depression. The model that I'll be telling you about we refer to as the social defeat paradigm. This is based on a large literature, decades old, in rats mainly, called the resident intruder assay. We've applied it to mice. This is work done by Olivier Bertone in the lab, who's now at Penn. And what Olivier did was the following. He took a C57 test mouse, put it in the cage of a bigger, meaner, retired breeder CD1 mouse. Those of you who watch the Discovery Channel know the ferocity of nature. Fighting is instantaneous, and it is very ferocious. We only allow the fighting to occur for a few minutes to limit physical injury. But for the rest of the day, the test animal is housed just with a screen separating it from the aggressor, still maintaining all other sensory cues. And we repeat that every day for 10 days with the test mouse exposed to a different aggressor. At the end of that 10-day period, we have induced in that test mouse a behavioral syndrome, the symptoms of which are defined here. So this is the equivalent of mouse DSM, Mouse Diagnostic Statistical Manual for Social Defeat, as valid as the definition of human depression. And the symptoms that are seen in these C57 mice are the following. The mice exhibit anhedonia-like symptoms, loss of pleasure, That actually is relatively easy to measure in a rodent. You can measure the degree to which they're interested in sex, food, sugar, high fat, and so on. They develop anxiety-like symptoms. It's a bit harder to know whether a mouse is anxious. That's simply referring to their interest in exploratory behavior. These animals are less interested in exploratory behavior. There's a hyperactivity at the hypothalamic pituitary adrenal axis. That simply means the mice secrete higher levels of glucocorticoids in response to various stimuli. They exhibit disrupted circadian rhythms, increased addiction liability, and what I'll be showing you examples of today, a metabolic syndrome and profound social avoidance. This is the way Olivier Berton devised our test of social interaction, placing the test mouse now in an open field. At one end of the field, there's a smaller cage which is either empty, that means no target, or another mouse present, that means target. In the absence of another mouse, the locomotor behavior of these animals is identical. But when a social target is placed in that cage, control mice spend a lot more time interacting with the social target. Mice are social, they're curious. However, the animals subjected to 10 days of social defeat show active social avoidance. It's not freezing, it really is actively avoiding the other mouse. We can quantify that simply by measuring the time the animals spend in what we call the interaction zone, and you can see a decrease in social interaction after 10 days of social defeat, a day after defeat, 4 weeks after defeat. In fact, it's essentially permanent behavioral abnormality. We've looked as long as 6 months. Importantly, this behavioral abnormality can be treated with chronic, not acute, administration of antidepressants. That's illustrated in this slide, work done by Olivier Berton and Nadia Tsankova in the lab. And what they found is that after 10 days of social defeat, now treating animals for 4 weeks with saline, we still see the persisting social avoidance. Single doses of imipramine and fluoxetine, two standard antidepressants, have no effect, but 4 weeks of treatment with these medications completely reverse this behavioral abnormality. Now, you might think that a mouse that's been beaten up 10 days in a row by another mouse, that it's normal to be socially avoidant. However, the important point is that these mice avoid their litter mates as well. So after this period of 10 days of social defeat, you could take the test mouse, rehouse it with its litter mates, and it remains avoidant for the rest of its life, essentially, which we think clearly places this as pathologic behavior. Other types of psychotropic drugs, for example, anti-anxiety drugs like Valium or diazepam, have no effect in the assay. Now, at about the same time, Chris Krishnan in the lab, an MD PhD student, noted that about a third of C57 mice do not exhibit these behavioral abnormalities. That's depicted in this slide, showing that control animals spend more time interacting with a social target than an empty cage, showing there's 2/3 of mice show the social avoidance that I've described already, and about a third of the mice fail to show that social avoidance. They show normal social behavior. We term these mice resilient. They seem to be resistant to this deleterious behavioral symptom, and interestingly, this resilience for social avoidance carries on to a resilience for other symptoms of social defeat. For example, the mice do not develop anhedonia. They don't develop a metabolic syndrome, but they still show the same level of anxiety-like symptoms. So the assay is interesting because it differentiates more depression-like behaviors from more anxiety-like behaviors. This illustrates what we refer to as the metabolic syndrome. This is work done by Mike Luter, who's now in Iowa. And what Mike found was that during a course of 10 days of social defeat, all animals subjected to the behavior lose a little bit of weight, but by the end of the 10-day period, their weights are pretty much back to control levels. When the animals are rehoused now under normal conditions, control mice and the mice that turned out to be resilient gain weight, as mice do, gradually and progressively over time. But it's the susceptible mice that gain weight much more quickly. They gain weight because they eat more. It's interesting, they eat more even though they enjoy eating less. And that increased eating and obesity is associated with a range of peripheral metabolic abnormalities that are somewhat akin to a pre-diabetic state. So this is interesting because we know in humans that people who are obese who are depressed are at increased risk for obesity. So this paradigm is social defeat, human depression? Well, of course not. I don't know what human depression is. However, is a clear set of consequences of chronic social stress, which a subset of mice are susceptible to and not susceptible to. And that provides an animal model now in which we can understand the neurobiology. Because of the behavioral abnormalities, our hypothesis is that changes in gene expression and play a key role in outcome. So let me just, for those of you who are not familiar, let me just describe what I mean by that. We know that the DNA double helix comprised in mammals of about 3 billion nucleotides. If stretched out linearly, that would be about 2 meters of DNA in each cell. Somehow that DNA is packaged into microscopic cell nuclei with an extraordinary degree of compaction and organization. The DNA is wrapped around octamers of histones, which form the unit of chromatin called the nucleosome, which are then further organized and compacted. And importantly, the state of chromatin in which a gene finds itself is a reflection of the rate of transcription of the gene. And we believe that this level of analysis is important for several key reasons. First, it will help us identify those genes that are altered in states of susceptibility and resilience. Perhaps even more importantly, from a mechanistic point of view, studies of chromatin provide the first ever indication of transcriptional mechanisms in the brain in vivo. All work prior to this point focused on changes in steady-state levels of messenger RNA expression, and any effort to understand the mechanism underlying those changes in mRNA expression relied on work in cell culture, even though we know what happens in cultured cells or even cultured neurons, is not an accurate reflection of what happens in the brain in vivo. By analogy with developmental and cancer biology, where certain epigenetic modifications are essentially permanent, perhaps some epigenetic modifications that occur in fully differentiated adult neurons are likewise long-lasting, underlying the stable behavioral abnormalities, and perhaps this could provide some insight into new treatment approaches. Let me give you a little bit more information focusing on this level of analysis. Now the DNA are these gold stripes wrapped around histone octamers, showing the two extremes of a spectrum of chromatin structure from active or open to inactive and condensed. In active chromatin, transcription factors bind to relatively open regions of DNA where they recruit co-activators, which would include, for example, histone acetyltransferases, which would acetylate N-terminal histone tails on nearby histones, all of which would then begin to recruit perhaps 100 or so proteins to form an active transcription complex. At the other extreme, chromatin is maintained in a condensed state by the actions of large numbers of repressor proteins. Examples would be histone deacetylases, which would remove the acetyl groups, certain types of histone methyltransferases that add methyl groups to certain lysine residues on these N-terminal tails associated with repression of gene expression, and DNA methyltransferases, which would add methyl groups to the DNA itself. The experimental approach we're taking is to overlay different levels of genome-wide data. So for example, we would start out with using RNA-seq, isolating regions of brain that I indicated earlier, identifying sets of genes whose mRNAs are induced or another set that are repressed. If these RNAs are regulated through transcriptional mechanisms, one would expect to see with an induced mRNA increased levels of chromatin changes associated with gene activation, or perhaps reduced levels of chromatin changes associated with gene repression. We would analyze these chromatin changes, again genome-wide, using what's called ChIP-seq, that's chromatin immunoprecipitation, followed by high-throughput sequencing that simply involves taking a brain region from a control and a stressed animal, fixing the tissue lightly to cross-link DNA with nearby proteins, fragmenting the chromatin, immunoprecipitating it with an antibody to, say, an acetylated histone or methylated histone, and then in the immunoprecipitate, those genes associated with that modification would be enriched, and those DNA sequences could be identified through high-throughput sequencing. And then we would overlay on top of those chromatin measures ChIP-seq genome-wide assessments of the binding of several transcription factors, which we know are also involved in chronic social defeat stress responses. For example, CREB, which is a pro-susceptibility transcription factor, or delta phos-B, which promotes resilience. So let me just give you now a progress report of some of the data that we've obtained. This is more recent work done by Rose Baggett in the lab, who applied RNA-seq 2 days after 10 days of chronic. This is showing the analysis of 4 brain areas. We actually have data from additional regions. I'll get to that in a few moments, from the same animals. Nucleus accumbens, the major reward center in brain, prefrontal cortex, basolateral amygdala, a major region of the amygdala complex, and the ventral subiculum, which is the major output of the rodent hippocampus. And some interesting patterns of gene regulation are apparent even from this genome-wide analysis. So for example, note that in most of the brain areas, nucleus accumbens is the most extreme, there's a lot more gene regulation occurring in the resilient animals than in the susceptible animals. And that was interesting to us because it suggested that resilience is not the absence of changes that occur in susceptible animals, but that resilience itself is an active process that's protective. In fact, one might formulate susceptibility as an absence of, or a failure of plasticity compared to a more plastic state of resilience. The other thing that becomes apparent is that the patterns of gene regulation differ from brain region to brain region. In some regions like nucleus accumbens or amygdala, by far many more genes are upregulated than downregulated, whereas in other brain regions, particularly prefrontal cortex and subiculum, you see the opposite type of pattern. One can take this large amount of data along with chromatin ChIP-seq data and deduce what types of upstream signaling pathways might be particularly regulated in states of resilience versus susceptibility. And I'm just showing you some examples arising from these molecular pathway analyses. Some of the results are surprising. For example, deducing that estrogen receptor mechanisms are particularly regulated in the brains of male animals subjected to defeat was not something we expected, but something that we're now seeing. Other results were more expected. For example, the implication of several inflammatory mechanisms would be expected based on increasing evidence in the clinic and animal models that peripheral inflammatory mechanisms and central mechanisms may be important in stress responses, the role of calcium and cyclic AMP pathways. But what I'll focus on for a few minutes now was the discovery also of very prominent regulation of Wnt beta-catenin signaling in this brain region. So we heard about this earlier today. I thought you guys would be interested in this for the role of these pathways in neurodevelopment, and now illustrating their critical role functioning in fully differentiated adult nerve cells. When we look at the pathway total, we see profound regulation of most proteins in this pathway. Just by way of review, for those of you who are not familiar with it, the pathway involves secreted proteins called Wnt, which act on frizzled receptors, and then which eventually activate the beta-catenin co-transcription factor through a variety of signaling pathways involving other proteins called disheveled directly, also indirectly through glycogen synthase kinase 3 beta. Disheveled inhibits GSK3 beta, which in turn inhibits beta-catenin. Hence, that's another pathway of beta-catenin activation. The different colors shown in this figure simply depict the different mechanisms of regulation found on our various ChIP-seq assays that I'm not going to describe for you today. What was found in our mouse model was a concerted direction of regulation. In susceptibility, most of these proteins were downregulated. This is in the nucleus accumbens, this major reward center of brain. Interestingly, we saw the opposite direction of regulation in resilient animals, with the pathway being upregulated. what's critically important in all studies of mental illness is try to validate studies in animal models in humans. The way we did this is collaborating with Carol Tominga and her colleagues in Dallas, where we obtained nucleus accumbens tissue from depressed humans and matched controls, and were able to show the downregulation of the signaling pathway in humans. So these are all correlative data. How do we establish causality? We can do that in a number of ways. This is work done by Matt Wilkinson and Carolyn Diaz in the lab. First, asking what the functional consequences are of a downregulation in disheveled beta-catenin signaling. The way we did this, shown in this slide, is by using a viral vector approach. In this case, herpes simplex virus vectors. We're expressing either a dominant negative mutant of disheveled or of beta-catenin. Not shown here, a wild-type GSK3 beta, because the direction of regulation is reciprocal. We can overexpress these dominant negative mutants in the nucleus accumbens selectively of adult animals and ask what happens to the animal's susceptibility to stress. What's shown in this slide is that those changes inhibiting this pathway, mimicking the molecular consequences of susceptibility, promote susceptible behavioral responses. So what's shown here is in response to a subthreshold degree of social defeat, just 2 days of stress exposures, normal animals overexpressing GFP in their nucleus accumbens still show the expected increase in social behavior when given the presence of a social target. But already after this very brief period of stress, animals with inhibition of disheveled or beta-catenin signaling show signs of susceptibility. We can further validate the effect of this overexpression approach by showing that injection of a viral expressing CRE into the nucleus accumbens of adult floxed beta-catenin animals similarly induces this increase in susceptibility. We can show the converse type of information, now overexpressing wild-type disheveled or wild-type beta-catenin, or conversely dominant negative GSK3 beta, and showing the opposite behavioral responses. Now in the context of a full course of 10 days of social defeat, the control animals expressing GFP show the expected social avoidance, which is completely prevented by overexpressing disheveled and beta-catenin, counteracting the stress-induced change, downregulation in those proteins. And although I focused in the data I'm showing you on this one assay of social behavior, we've seen the same types of regulation in other behavioral assays, specifically in a measure of anhedonia or sucrose preference. Now, this would suggest that pharmacological means of activating this pathway might be There has been a great deal of interest in the pharmaceutical industry in developing GSK3 beta antagonists. However, that has not proven to be a fruitful approach. These inhibitors are quite toxic, as developmental biologists might presume, and certainly beginning with developing drugs that are Wnt agonists would be particularly hazardous. So the approach that we've been interested in is to look for more downstream effectors of this pathway that might be more enriched in nerve cells, perhaps even enriched in nucleus accumbens, which could then give us insight into new druggable targets. So the approach we've taken is to carry out ChIP-seq on beta-catenin in susceptible and resilient animals, and I'll just give you one example of some of these data. This is, again, work by Carolyn Diaz in the lab, and I'm just showing you one slide to kind of illustrate where this work is going. The pie charts illustrate the distribution of beta-catenin binding genome-wide. About half of the binding is associated with gene regions, either promoters or gene bodies. And what's clear, even at a genome-wide level, is that there's a profound reorganization of beta-catenin binding in gene regions in susceptible versus animals. I think you could see that in susceptibility, there's reduced binding sites of beta-catenin at promoter regions and gene body regions, and a dramatic increase in beta-catenin binding in states of resilience. When one analyzes then these, the induced beta-catenin binding sites in resilient animals versus susceptible or control animals, one can identify biochemical pathways that are regulated by beta-catenin or RNA and come up with very rich data sets of biochemical pathways that are modified. I don't expect you to be able to read that slide, but we are already pursuing several of these downstream targets as potential mediators of the pro-resilience action of beta-catenin. Let me give you another example of how we've mined these genome-wide data sets to understand mechanisms of resilience. This is now looking at old microarray data from the ventral tegmental area. The VTA is the site of dopamine neurons that innervate forebrain regions. And we noticed that the VTA, like the nucleus accumbens, showed this dramatic preference for gene regulation in a state of resilience compared to a state of susceptibility. And what was apparent in comparing these data sets is that among the genes that were induced uniquely in resilient animals and not in susceptible animals were a series of potassium channels, including several subunits of what are called KCNQ channels. And this gene discovery data was the first indication we had that perhaps susceptibility versus resilience was being controlled by the intrinsic excitability of VTA dopamine neurons. The prediction would be, through an induction of potassium channels in VTA dopamine neurons in resilient animals, that these resilient animals would be less excitable. That's not exactly what we found. What we found instead was that the susceptible animals showed a significant increase in intrinsic excitabilities that was normalized in resilient animals by the induction of potassium channels. These are data from, but the same phenomenon is observed recording from these nerve cells in vivo. Now, importantly, this induction of excitability of VTA dopamine neurons is specific for those VTA dopamine neurons that project to the nucleus accumbens. I'll describe in the next slide how we made those determinations. But importantly, when we focused on another subset of VTA dopamine neurons, those that project to the medial prefrontal cortex, for example, we saw a very different pattern of activity of these nerve cells. We were able to show that these nerve cells showed a reduction in excitability in susceptible animals, so the opposite phenotype. This is work initially done by Ming-Hu Han in the lab, and more recently by Junli Cao and Dipesh Chowdhury. So again, all correlative data in order to directly implicate a change in excitability of VTA dopamine neurons in susceptibility versus resilience, we turn to optogenetic tools. And based on the data I indicated, it was crucial to target select microcircuits of VTA dopamine neurons, those projecting to nucleus accumbens versus those projecting to medial prefrontal cortex. So the way Dipesh and then also Jesh Walsh did this experiment was the following. They injected a pseudorabies virus expressing Cre recombinase into the nucleus accumbens. This was a deficient pseudorabies virus. It is not capable of jumping synapses, but it does get picked up by all neural elements within an injected brain area. So not only would nucleus accumbens cell bodies express PRV Cre, but all the nerve terminals that innervate the accumbens would pick up the virus, transport it retrogradely back to their cell bodies, no further transport, but those cell bodies too would become Cre positive. And then we could inject a channel rhodopsin expressing AAV vector where the channel rhodopsin expression is Cre dependent into the VTA, as depicted in this slide, therefore only infecting those neurons in VTA that project to the nucleus accumbens, 98% of which are dopaminergic. And what we found in this study is the following. Now with a subthreshold, again, measure of defeat, which in control animals is not enough to produce a behavioral abnormality. These animals, control animals expressing GFP, still spend more time with a social target than without. But animals now exposed to excitation of these VTA dopamine neurons, essentially mimicking the effect seen in susceptibility renders these animals far more susceptible. The pattern of firing of these VTA dopamine neurons is absolutely essential. Only phasic firing has this effect. The same number of nerve impulses just delivered in a tonic pattern has no effect. The same data were obtained not only with social avoidance, but also with sucrose preference, exciting these neurons, inducing a deficit in sucrose drinking. We can obtain the converse type of information, now expressing halorhodopsin, which inhibits the activity of nerve cells. Halorhodopsin, after 10 full days of chronic defeat, quieting these VTA dopamine neurons, immediately reverses the depression-like behavior, both in the social avoidance test and in the sucrose preference test. We can then ask what happens when we control bidirectionally the activity of the other subset of VTA dopamine neurons, those that project to prefrontal cortex. And by the way, there's virtually no overlap between these two subsets of VTA dopamine neurons. Now, and Jess injected the pseudorabies virus Cree into prefrontal cortex, injected the AAV Cree-dependent vector expressing channel rhodopsin or halorhodopsin into the VTA, and asked what happened. We found no effect when we turned these nerve cells on, but when we turned these nerve cells off, when we inhibited their activity with halorhodopsin, mimicking what happens to these neurons in susceptible mice, we promoted susceptibility after subthreshold levels of defeat. Now, interestingly, in this case, we only saw this pro-susceptible-like behavior in the social interaction test, no effect on sucrose preference. Now, all the data that I've been presenting thus far are looking at short time points after defeat. All of the genome-wide data I've shown you to this point is carried out at 2 days after 10 days of social defeat. I've already mentioned that the behavior, so we've more recently turned to RNA-seq and ChIP-seq at these longer time points. And work done recently by Rose Baggin in the lab characterized the same 4 brain areas shown here. Now, 1 month after the last social defeat stress, animals have been rehoused into normal housing. And what's dramatic is very different of gene regulation observed at this longer time point. No longer does the nucleus accumbens stand out as the most dramatically regulated with a preponderance of gene regulation in resilient versus susceptible. But what stands out at this steady state time point, this is looking at mRNAs that are altered at a steady state level a month after defeat, you see a dramatic regulation, predominantly gene induction, in the amygdala. It's interesting because the amygdala is particularly implicated in emotional learning, so we're very interested in looking at those responses. We then carried out an additional data set in another group of animals, analyzed 4 weeks after chronic social defeat stress, where the animals were then re-exposed to another stress. And again, found still different patterns of gene regulation. What's particularly dramatic here then is different global patterns of gene regulation arising in these different brain regions. Gene regulation in susceptible animals being quite apparent in nucleus accumbens, but gene regulation in resilient animals more apparent in prefrontal cortex and so on. When you drill down into these data sets, 2 days after defeat, a month after defeat, with a stress challenge, you see genes that represent all imaginable patterns of regulation. Genes that start out abnormally regulated, that persist abnormally regulated a month later. Genes that are normal right after defeat that become abnormal in more of like an incubation type pattern, as well as genes that start out abnormal and then revert to control. Presumably, these various patterns of gene regulation are reflecting changes at the chromatin level. So I'd like to end by just showing you some data, giving you an indication of some of the chromatin data that we're obtaining. This is work done again by Matt Wilkinson, more recently by Jian Feng and others in the lab, looking at ChIP-seq for 2 major histone and transcription factor modifications. We focused on one particular, here I'm telling you about one particular form of repressive histone methylation. Let me give you the key. This stands for histone 3 lysine 9 dimethylation. This is a major form of repressive histone methylation. It also, in our ChIP-seq data, is one of the best predictive chromatin measures of changes in mRNA expression. We've also found, work by Herb Covington and Ian Mays in the lab, that the histone methyltransferase that catalyzes this particular form of repressive histone methylation, G9A, is robustly involved in controlling susceptible versus resilient responses. The other mark I'm telling you about is phospho-CREB. Phospho-CREB is the active form of CREB. I mentioned earlier that CREB is a mechanism of susceptibility. A large literature from our lab and other labs has shown that after chronic stress, CREB is induced in nucleus accumbens. We showed it's primarily induced in susceptible animals and with many measures drives a state of susceptibility. What I'm showing you now here is the changes in these two marks a month after social defeat, and I'm only showing you genes that show coincident changes in mRNA expression based on our RNA-seq data. The value of overlaying these different platforms is that it vastly improves the accuracy of the data, drives false negative, false positive discovery to very low levels. And what show is that there is common regulation after chronic treatment with an antidepressant that restores normal behavior compared to the resilient mice that maintain normal behavior despite the exposure to chronic stress. So that for both of these marks, the patterns of regulation seen in susceptible animals are largely not seen after chronic treatment of susceptible animals with imipramine or in resilient animals. And in fact, one can compare these data sets under these two conditions, resilience versus successful antidepressant treatment, and show that there's quite a bit of overlap in the genes that are regulated under these two conditions. This is a very interesting finding to us because it suggests that one of the ways in which antidepressants may be working is by inducing in sick individuals some of the same mechanisms that occur naturally in those individuals that are inherently more resilient. And it suggests that one of the ways in which we can improve antidepressant drug discovery, in addition to look for ways to undo the bad effects of stress, is to look for ways to induce a state of resilience in individuals who are not inherently resilient. And in fact, our example of induction of beta-catenin or induction of potassium channels are two examples of these pro-resilient mechanisms that we're very interested in now in taking in a drug discovery path. So with that, let me just conclude by saying that I hope I've convinced you, despite the nihilism I expressed at the beginning of the talk, of the difficulty in studying the biological basis of mental illness, that it is, with the judicious use of animal models, possible to understand something about how the brain reacts positively and negatively to periods of chronic social stress, and learn from that something about what makes an animal susceptible or resilient, as well as understanding how antidepressant medications act after chronic administration. We're now dramatically expanding assays in the following types of ways. We have a number of situations where we can take animals early in life and increase or decrease their inherent rate of resilience or susceptibility in a lifelong way. For example, taking C57 mice, exposing them to certain forms of maternal separation at a particular time in development renders those mice more susceptible for life. Presumably that's through stable changes in gene expression, which we can now identify with the methods that I've described to you today. We're correlating these data in animal models with genome-wide investigations of homologous brain regions of tissue from depressed humans. I gave you an indication of that from our work on the Wnt disheveled beta-catenin pathway. And we're also looking at other chronic stress models. We do believe that changes at the epigenetic level are one important way in which stable changes in gene expression are induced and maintained in nerve cells in response to chronic stress. And we're very interested in looking for ways to take advantage of these data to improve our diagnosis and treatment for depression. So again, thank you very much for having me. I'd be happy to answer any questions.
Eric, I know you said those forced swim tests and also the other tests are sort of problematic. Have you tried after this chronic social defeat, then put the mice or the defeated mice to those tests to see whether there's any truth to those tests?
Yes. So the question is, what happens after a period of chronic social defeat stress in susceptible animals? What is the response of those animals in a forced swim test, for example?
Right, for example, or
it's very variable. In general, we see no effect. But one of the things that we see in most cases when we expose animals to stress and then test them in the forced swim test, the animals are more robust in the forced swim test. And I think that makes a lot of sense. In other words, they're antidepressant-like in the forced swim test after stress. I think that makes a lot of sense because it's likely that the prior stress is almost like special forces training for the rodents and makes them tougher.
Okay. And the second question...
So again, it further emphasizes the notion that the forced swim test and these other rapid assays are not accurate reflections of the hedonic state of an animal.
Completely agree. So the second question is for the resilience part. You mentioned DVL-B is also pro-resilience. What's the relationship between DVL-B and beta-catenin? Do you see any relationship there?
We've looked extensively at that and have not seen any relationship. If we manipulate beta-catenin, we don't see any obvious changes in expression in delta-B. If we manipulate delta-B, we don't see any obvious changes in activity or expression levels of beta-catenin. There are AP1 sites on many of the genes in the beta-catenin, disheveled beta-catenin pathway, but we don't see them as being very functional in the nerve cell types that we've looked at. Conversely, there are some TCF, LEF sites in the FOSB gene, but again, that doesn't seem to be functional. So we think they're independently acting.
So it's sort of parallel pathway.
Yeah.
And finally, the dopamine signaling, the phasic versus tonic.
Yeah.
I want you to elaborate a little bit on that. So how do you think of the difference between phasic and tonic? Especially consider, I remember Diceral's published a paper seems to have a slightly opposite conclusions on the effect
Yes,
of phasic
yeah.
versus tonic.
So that's really two questions now.
Right.
So the first question is, it's known that VTA dopamine neurons show two patterns of firing in vivo. More tonic pacemaker firing, that's due largely to the intrinsic activity of these cells, as well as phasic firing, which is more a reflection of the regulation, activation of those cells by incoming glutamatergic afferents. The phasic firing is far more effective at releasing dopamine. It is also far more effective at releasing BDNF, which is expressed by VTA dopamine neurons. So in fact, we believe that in the context of chronic social defeat stress, what's the critical factor in inducing of the increased firing rates of dopamine neurons inducing susceptibility is BDNF. And we've shown that in a number of ways. We have another paper coming out that does it more definitively, really showing that the consequence of phasic firing that's crucial for susceptibility is BDNF. Okay. So that you also asked Kay Tai, now at MIT, who is with Carl Diceroth at Stanford, had a companion paper in Nature with our paper, basically showing the opposite effects. So with chronic mild stress, they found that increased firing of these dopamine neurons is antidepressant-like, whereas we showed increased firing was pro-susceptibility. We have a lot of theories why that might be. Carl and I have talked about it extensively. I think it emphasizes a few things. First of all, the effect of these dopamine neurons is known in a large literature to be highly context-dependent. And we believe that there are different features of these stress models that have to be taken into consideration. Again, really highlighting what I said at the outset, that there's no such thing as a single type of depression or a single type of stress. Social stress, we think, is much more severe than a chronic mild stress paradigm. So we think in states of mild stress, more dopamine activity may be antidepressant, protective. But in states of extreme stress, what we think is happening is that the excessive phasic firing of these dopamine neurons through a BDNF mechanism is enhancing plasticity to the point in these animals that they're becoming behaviorally inflexible. And in the context of social defeat stress, that's not good.
Okay, thank you.
Yeah.
You mentioned that in humans, metabolic syndrome and depression tend to be mutually reinforcing.
Mm-hmm.
So if you were to compare the RNA-seq and chip-seq data of the susceptible animals with, say, the similar data for an animal model with metabolic syndrome,
mm-hmm.
would you expect to find interesting differences and common?
Yes, absolutely. It's a great idea. It's something we're very interested in doing. We haven't done it yet. But again, the thing that's important is that in humans, there are subsets of depression that are not associated with obesity. Some people who are depressed don't eat and lose weight. And what we think would be, and conversely with obesity, not all obesity, probably majority obesity is not due only to overeating. But for the slice of obesity that is due to overeating and the slice of depression that's associated with obesity, we would expect to see common molecular pathology in these limbic structures. And it's something we're very interested in looking at.
So you mentioned in the first beginning a big advance, the deep brain stimulation. If those resilient mice also respond to deep brain stimulation?
Well, what we have shown is that deep brain stimulation, we didn't do deep brain stimulation. We've used optogenetic tools focusing on the homologous regions that have been shown to be antidepressant in human, medial prefrontal cortex and nucleus accumbens. And we've been able to show that optogenetic activation of both of those regions is antidepressant-like in our social defeat model. That's focusing on the susceptible animals. We have not seen a change in behavior when we do that, when we employ those tools in the resilient subgroup. One of the important things just to add about the optogenetic analyses is that when surgeons implant DBS electrodes in a human, it's very unclear what functional effect a metal electrode is having when it's stimulating a collection of brain tissue. It could be stimulating or inhibiting cell bodies, terminals of several types, glial cells, and so on. With optogenetic tools, we can target select cell terminal populations and dissect out the circuitry more effectively. Yes?
So nucleus accumbens contains this D1 and D2 subpopulations, which corresponds to presumably the appetitive and aversive emotional response. So have you tried RNA-seq on these two different populations separately? And also for this beta-catenin pathway, do you have any speculation which one of the D1 and D2 population it's predominantly acting on?
Yes. We are now carrying out RNA-seq on D1 and D2 neurons. It took quite a lot of effort to develop the tools to be able to carry out RNA-seq on such small numbers of cell populations. Nucleus accumbens is big for Drosophila, but small for a mouse. It's about a pinhead in size. And isolating by FACS D1 versus D2 cells doesn't derive that much tissue, but we now have it working very well. And interestingly, we now know that the roles of these different susceptibility resilience factors do seem to be playing very cell type specific actions. So for example, the pro-resilience effect of the beta-catenin signaling is through D2 cells. And we've been able to show that in several ways. We can show that the induction of beta-catenin signaling in nucleus accumbens in resilient animals only occurs in D2 neurons. And when we use our viral tools in D1 Cre or D2 Cre mice with viral vectors that are dependent, we can show that beta-catenin only exerts an antidepressant or pro-resilient effect when we overexpress it in the D2 cells.
So I have a couple of questions. One is, have you ever taken your populations of susceptible and resistant animals and interbred them and maintained them over generations to see if those epigenetic modifications might be maintained, if you will, in the groups?
Right. So one factor that I swept under the rug is that, although I did mention it, all the experiments I talked about were on C57 mice. Now, presumably they're genetically inbred, and I think the numbers and the magnitude of differences we see suggests that the difference in phenotype, behavioral phenotypes, are not due to smaller genetic variations within an inbred strain like copy number variation. I think it would be highly unlikely. And the RNA-seq actually doesn't bear that out. We haven't committed to do breeding of the two subgroups, but we did do the following experiment. We took a group of C57 mice without knowing whether they were resilient or susceptible. These are latent traits, and we haven't yet identified a way to distinguish which C57 mouse is going to become susceptible or resilient before the stress. But we took a group of mice and bred them to normal females. We subjected them to stress and ascertained which mice were susceptible and resilient. We then looked at the offspring of the mice generated before defeat and found no difference in rates of susceptibility or resilience in offspring of mice that were destined to be one or the other. So that was unfortunate because we thought that there might be a pre-existing epigenetic state that could potentially be transmitted, but it's not. Does that answer your question?
Yeah, I think so. I mean, I don't know much about whether epigenetics, if there were marks that were due to some environmental factor that they might be maintained or not over generations. I don't know, but the inbreeding is obviously clear, and it seems unlikely there would be another genetic source.
You know what? There are a few small examples where true epigenetic inheritance has been demonstrated. But I'm skeptical that it occurs in the context of what I'm describing today. We did another experiment where we took susceptible mice and bred them to normal females, and then collected sperm from the same mice and impregnated other females with IVF from that sperm. And what we found with normal sexual reproduction, that there was a dramatic transmission of behavioral susceptibility. Really shocked me. However, the transmission with IVF was extremely subtle, suggesting that most of that transgenerational transmission is probably behavioral. Right? And that's more plausible to me in some ways. We still don't know what types of mechanisms could be transmitted. Again, it's a high bar. There have been reports in the literature of the histone acetylation at the BDNF gene is altered in sperm. I mean, that's really not plausible as a mechanism for transgenerational transmission of a stable behavioral abnormality. Anyway, that's a
cool experiment. I'll ask you the other stuff later.
Okay.
Well, it's just a quick question. So have you seen any correlation between the susceptibility versus the level in social hierarchy of the animal?
Yeah, good question. So again, part of our effort in ascertaining if there was a pre-existing detectable behavioral state prior to the stress that would differentiate those animals that were going to become susceptible or resilient, part of that was analyzing dominance hierarchies, maternal care to the different pups, and we could find no correlation at all between all those measures and susceptibility versus resilience.
Has anyone tried to derive IPS from depressed patients? And do you think will this epigenetic change kept in these cell models?
I'm sorry, so say that again. You're talking about LPS, lipopolysaccharide?
Has anyone tried to derive IPS from depression patient, human patient? And will this epigenetic change be kept, in these cell models?
So we're looking at epigenetic changes in human brain, but it's hard you can't do that in a patient, obviously. And so the only tissue that's accessible in a patient is their blood or skin. And we have carried out some initial genome-wide ChIP-seq assays on skin and blood of patients, and not surprisingly, the patterns of gene regulation are very different from what you see in brain, of course, because even in brain, what we see are highly nerve cell to nerve cell dependent changes. So I'm not optimistic that anything we're going to see peripherally is going to be that predictive of what's happening in the brain.
I might have missed it, but if your expression profiles are predictive, if you had an experimenter that was blind to the behavior of the animals and only knew the expression profile, could they accurately predict resilience versus susceptibility?
We haven't done the experiment in the way you're describing, and it's a very good idea to look at, because we have now several generations of gene expression and ChIP-seq data, and so that we would have many cohorts to examine, teaching cohort and then separate cohorts to see if the predictive power of patterns hold through. So that's something actually that we would like to do. What I can say clearly is that there are some changes that we have found that are large enough in magnitude that clearly differentiate susceptibility and resilience. The firing rate of VTA dopamine neurons is one of the most dramatic, and I didn't point it out in a small panel. The correlation between depressive-like behavior and the firing rate of VTA dopamine neurons is extraordinary. Levels of delta phos-B as a transcription factor in nucleus accumbens, likewise, very robust, very differentiating. So I know that there are a few individual molecules. Presumably, if we did genome-wide analyses, we'd be able to find combinations of regulation that would be even more predictive. So I'm optimistic that we could do that.
So as you mentioned that the VTA nuclear accumbens pathway, the activity of this pathway is increased in the susceptible mice, and I'm very curious about the activity of the whole nuclear accumbens. Have you tried to inactivate the whole nuclear accumbens to see the behavioral performance of these animals?
Yes. This is also work done by Mary Kay Lobo and her colleagues in Maryland now, a former postdoc in the lab. And we and others have tried to use optogenetic tools to activate the nucleus accumbens or specific cell types and so on. And in general, what is interesting is that it's not so much the intrinsic firing of the nucleus accumbens nerve cells that seems to be the most potent determinant of susceptibility or resilience. What seems to be much more capable of controlling those behavioral patterns is the strength of the glutamatergic innervation of those regions from various afferent limbic regions like amygdala, ventral subiculum, prefrontal cortex, and so on. And what we've seen so far is very clear input-specific differences in the consequences of activation of glutamatergic synapses from one brain area versus another on nucleus accumbens nerve cells and controlling those behavioral responses.
Whether the beta-catenin signaling pathway is conserved in different animal models of depression, and whether this pathway can be regulated by dopamine or BDF in the VTA.
We have evidence that the activity of the beta-catenin pathway in nucleus accumbens neurons is regulated by BDNF. That study is not complete yet, so dopamine might also regulate it. How that regulation occurs is not known. We haven't yet delineated the signaling pathways from the BDNF to beta-catenin itself. But it does seem to be able to, BDNF exerts a repressive effect on activity of beta-catenin signaling in D2 neurons.
In your only-seq data from 2 days and 10 days after social defeat, presumably same group of, similar group of genes probably activated or downregulated.
Mm-hmm.
So I wonder if that's the case you see. For example, the beta-catenin pathway, do they show up in both?
So again,
data?
we've seen several patterns of temporal regulation. Beta-catenin pathway genes are examples of genes that in resilience are induced right after social defeat and stay induced for several weeks. So that's an example of a set of genes that show consistent, stable regulation over time. Other sets of genes that are altered at early time points revert to normal within a few weeks or even earlier in many cases. A third subset of genes may be the most interesting. These are the genes that seem to show normal levels of steady-state expression immediate after defeat, but for which abnormal regulation becomes apparent in an incubation-type manner during withdrawal from social defeat. And we're very interested in drilling into those individual genes that show those patterns and understanding the molecular mechanisms underlying the temporal regulation. Thanks.